microscopic units or cells. Therefore, this most primitive that accounts for differences in the bulk of individual animals. This is as true for this sponge as it is for an elephant or a mouse. We can demonstrate the relationship between cellular structure and bulk by using egg white. Egg white has a consistency much like living protoplasm. A large amount of egg white cannot maintain a constant shape. But by whipping the egg white, we form cell-like masses of air and albumin. Roughly, these masses may be compared with protoplasm, which has been partitioned into cells and enclosed in membranes. Unlike plain egg white, the air and egg white mixture can hold almost any form, especially when supported by a skeletal structure, such as excelsior. This analogy applies to all many-celled animals. There is another, even more compelling reason why animal bodies larger than protozoans must consist of many cells, not just larger masses of protoplasm. Only small masses of protoplasm can receive sufficient oxygen and dispose of wastes fast enough to support life. We can demonstrate how this works. Here is a cube representing a cell. The ingoing arrows represent oxygen entering the cell. Oxygen can only enter through exposed surfaces of the cell. Furthermore, it can only diffuse into the cell at a given rate of speed. The outgoing arrows represent wastes leaving the cell. Again, diffusion is so slow that a large mass of protoplasm simply could not rid itself of wastes fast enough to stay alive. But if this larger mass is divided into many smaller units, with spaces between them, much more surface area is exposed. With more exposed surface, sufficient diffusion can now take place. We may consider this need for many cells in animals larger than protozoans quantitatively by using these model cells. This large cube is 3 inches on a side. This small cube is 1 inch on a side. "27 of the small cubes equal the size of the one large cube. By carefully removing a thin paper coating representing the cell membrane from the large cube, we can readily observe the total surface where diffusion into or out of this mass could occur. In the same way, we can remove the surface coating of one of the 27 small cubes. Finally, by comparing the total surface area of all the small cubes with that of the one large cube, we can see the enormous increase in surface area relative to volume, made possible by splitting the large mass. The increased surface area, the spaces between the cells, and the shorter distance that oxygen and wastes now have to travel make possible a body plan that permits growth beyond the size of protozoans. In all animals made up of many cells, except the colonial protozoans, certain cells are adapted to perform specialized tasks. Such specialization leads to greater efficiency and the probability of exploiting new sources of energy. How does the body plan of a sponge bear out this generalization? Essentially, the sponge is a vase-shaped sac with a large opening at the top and microscopic pores perforating the sides. Food and water enter these microscopic in-current pores. Because of the small size of these pores, the sponge can feed only on microscopic organisms. Water passes into the main cavity, then leaves by way of the large excurrent opening, so there is a continual flow of water through the sponge. By using a dye, we can see the movement of water through the body of a living sponge. The dye moves toward the wall, and disappears into the pores of the sponge. We can see the dye particles emerging from the large excurrent opening. If these had been food particles, the sponge would have retained them. The principal types of specialized cells in the sponge are identified in this drawing. The sac is covered and protected on the outside by flattened covering cells, which fit together like the tiles in a mosaic. Through the pore cells, water is drawn into the sponge. The large internal cavity is lined with collar cells. The word collar refers to the delicate collar of protoplasm that encircles the free end of each cell. This free end also bears a long flagellum, whose base passes through the collar. It is the beat of the flagella that creates the water current which passes through the sponge. As the water passes through the sponge, the collar cells capture food organisms. Between the covering cells and the collar cells are moving mesenchyme cells. They receive partly digested food particles from the collar cells, complete the digestion, and carry the digested food from one part of the sponge to another. A function of some of the mesenchyme cells is to secrete needles of calcium carbonate. These needles are called spicules. The spicules form a framework which supports the soft cellular mass, much as the excelsior supported the egg white in our earlier experiment. We can detect the presence of calcium carbonate, or limestone, in the skeleton with dilute hydrochloric acid. The acid dissolves out the skeleton of the sponge. Here are different kinds of spicules seen under the microscope. Mesenchyme cells also have a part in the sexual reproduction of sponges. Some cells enlarge with reserve food and become eggs. Others enlarge and become sperms. Sperms are brought inside the sponge in its water current. Eggs, such as the one shown here, develop into flagellated larvae. The larvae escape from the parent and swim about until they find a place to become attached. Each surviving larva then grows into a young sponge. Sponges may also reproduce asexually by budding and branching. Sometimes buds are torn off and grow into complete individuals. This ability to regenerate living parts is more pronounced in the sponge than in higher forms of animal life. This is a freshwater sponge. Like most freshwater forms, its method of reproduction is somewhat different from that of saltwater forms. Its mesenchyme cells build up an asexual reproduction unit known as a gemmule. Here is a gemmule seen under a microscope. It consists of a mass of food-filled mesenchyme cells surrounded by heavy protective coats strengthened by spicules. Such gemmules can survive drying and freezing. Under favorable conditions, the sponge cells emerge through a thin spot in the gemmule coat, aggregate in a small mass, and grow into a new sponge. Sponges seem to have changed little during their 500 million years of evolutionary history, but some variations have developed. Most evolutionary changes have been in the system of canals and the skeletal structures. Sponges have few enemies. One is the nudibranch. Some browse on living sponges. More dangerous are other living forms that grow over sponges like hydroids or algae. Such forms of life can choke some sponges by covering up their intake pores. Silt or mud may also cover the intake pores. Some types of sponges have adapted to such conditions so that the growth of algae is not harmful. In fact, it may even protect the sponge from intruders. Sponges do not respond in any conspicuous way to cutting. This apparent immobility does not mean that the sponge is totally incapable of movement. There is some movement, but it is extremely slow. A stimulus does not affect more than a very restricted part of the sponge's body. Occasionally, one finds what appears to be a moving sponge. A closer look shows it is a sponge growing on a crab. The sponge serves to disguise this crab. Hermit crabs sometimes use sponges as shelters. There are more than 5,000 different species of sponges, grouped according to the composition of their skeletons. Some have skeletons of calcium carbonate. Some have skeletons made of glassy silicate, like this Venus's flower basket. Others consist of silicate combined with spongin, a horny fibrous material. Still others are made of spongin alone. These are useful as bath sponges. Such sponges have commercial value and are collected by divers. Trampling and washing breaks up and kills the living cells, leaving the skeleton behind. From time to time, they are checked to see if they are well cleaned. Finally, the soft skeleton of spongin appears. Under a microscope, we can see the fine structure of this skeleton, which appears as a three-dimensional network of fine threads. This network provides strength, elasticity, and a framework for the cells. Under the microscope, too, we can see how water moves through the skeleton of the sponge. The water can be removed with the slightest pressure. This makes the sponge especially useful in washing. Biologists have long been interested in the sponge's remarkable powers of regeneration. We can demonstrate what happens when we take a piece of living sponge and disassociate its cells. The cells are squeezed through a bag of fine bolting silk into a dish of seawater. A drop of this suspension, when examined under a microscope, shows that it consists of a large number of separate sponge cells. The cells are well disassociated. When they settle in the bottom of the dish, they begin to wander about in an erratic manner. This time-lapse photomicrography shows individual cells joining together into small groups of 6 to 20. Sponges as large as a pea are formed. This unusual development is significant because the cells have crept together by themselves. This mass contains various cell types, such as collar cells, covering cells, and pore cells. Such experimental work on sponge regeneration not only provides information on the nature of the sponge's cellular structure, but may also help provide additional knowledge about the mechanisms of wound healing and tissue grafting in other animal bodies. The sponge illustrates the development of multicellularity in animals, and some of the ways in which the body plan of this primitive animal fits it for survival. Although the sponge is interesting because it illustrates the cellular level in animal structure, a stage no longer found among other many-celled animals, it is little more than a side issue in the trend of evolution.